Hi guys, today we are going to cover two things. First off, we're going to have a review of some pipetting techniques, and we're going to do that through also preparing a Bradford standard curve to determine the protein concentration of an unknown protein. So for today, our unknown is going to be a GFC purified fraction from the purification we did a couple of weeks ago. And so things that you should be paying attention to would be how I'm holding the tubes, how I'm using the pipettes in particular, and the general organization of the workspace. Workspace organization is really going to help you as you do your experiment, as you set up your assays, and it's going to help you keep track of your different samples and prevent you from making needless errors. So here we go. The first thing that we need to do is get gloved up. We need to be very aware of personal protective equipment, of course, in the lab. So we're going to get a pair of gloves, wearing a lab coat, wearing eye protection. Hair is tied back if you have long hair, and we're going to get gloved up here. Notice the fit of the gloves. These gloves should not be loose on your hands. If they are loose, little bits of the glove are going to get caught in tubes as you try and close them and so on. Okay. We need to identify our samples. So here we have, first off, our BSA standard that we're going to use to make our standard curve, and then our protein that we're going to determine the concentration of. And so this is labeled GFC because it's our GFC fraction. First thing I want to do is make a series of standards to make my standard curve. So I'm going to use my standard for this, and you'll note it's labeled with the concentration. So the concentration of my, my stock here is 1 mg per mil, and this is a BSA standard which is used for protein determination quite commonly. So I'm going to get some empty Eppendorf tubes, some 1.5 ml microfuge tubes here, in order to make my standards. And my standard curve is going to have 5 points. The highest concentration will be 1 mg per mil, which is what my stock is. So I then need 4 more microfuge tubes to make up my the rest of my standards. I'm going to make the standards by doing a 1:2 serial dilution. The volumes that I use for this are somewhat irrelevant. I need to keep in mind how much of the standard will I use in my assay. And I'm going to be using only 25 microliters in my assay. So I can, as long as I have more than 25 microliters made up in these tubes, it doesn't really matter what volume it is. I'm going to make up 100 microliters of each standard. I'll do that by pipetting first my diluent into each one, and then by doing serial dilutions from one tube to the next. You could do this using 500 microliters, using 300, or whatever volume that you prefer to pipette. So because I want to pipette 100 microliters, I need to select a pipette. So I have 3 pipettes here. I have my P1000, which looks blue on the top. I have my P200 and my P20, both of which are yellow. Make sure that you check the tops of them so that you've selected the correct pipette. So I need my P200, which is this one, so that I can pipette 100 microliters. It's not appropriate to use a P1000 to pipette 100 microliters if you have a P200 available. So in order to determine how to wind the pipette to the correct volume, you're going to have a quick look at the display here. There's 3 digits displayed, and to know what these digits should say, the first thing that you're going to ask yourself is, what is the maximum volume this pipette can aspirate? And this is my P200. So when this is wound to its maximum, it will say 200. I want 100, so I will just wind this to 100. And so I'm just going to wind this here. Do take the time to ask yourself, what would the maximum volume look like? Especially if you're pipetting a lot of things, it's really easy to get confused and just pick the wrong pipette, or wind it inadvertently to the wrong volume. And usually if you get it wrong, you're out by a factor of 10, which is huge. So you don't want to do that. Okay, so I'm going to start off by pipetting my diluent. My diluent is going to be bicene 0.03 molar at pH 8.5. The reason that I'm using this as a diluent is because this is actually the buffer that my GFC unknown protein is in. So that's an appropriate diluent to use. So you want to make sure your workspace is set up organized correctly. Let's get this out of the way. Don't need this Sharpie right now, so we'll get it out of the way. If you're right-handed, you're going to be pipetting from right to left. If you're left-handed, you're going to be going this way, obviously. Start by what you want to do, moving your work from right to left across the order that you need to do things. That will prevent you from dragging your sleeve across your samples, your very, very valuable samples. So I'm going to take my pipettes here and put my yellow tip on, make sure it's on securely, and then I'm going to depress until I feel that first bit of resistance here. Make sure that you don't go all the way down to the end, that's inappropriate. I'm just going to go to the first resistance, insert into the liquid. And this is a relatively small volume. I don't know if you can see very clearly there. 100 microliters is not very much. So I'm going to pick up this tube and I'm going to make sure that I can see exactly what I'm pipetting into the tube and that every drop goes in. 100 microliters is a small amount, and if you miss a drop going in, that is a significant volume. You could end up being out by, 5%, and that's error that you can avoid pretty easily. I can use the same pipette tip because, number one, I'm pipetting the same thing into every tube, but more importantly, there's nothing in these tubes, so there's no opportunity to cross-contaminate my buffer with something that was already in these tubes. Okay, so I have pipetted 100 microliters into each of those. Now, I'm going to be doing a 1:2 serial dilution. If I have 100 microliters in here, I'm basically looking to halve the concentration of my BSA. So I'm going to take 100 microliters of this, which is 1 mg per mil, put it into 100 microliters of diluent, and thereby halving the concentration of this. So my new standard is going to be 0.5 mg per mil. Now, something that I just noticed about my tubes is that I don't have them labeled. So I'm going to actually just take a minute, I'm going to close these tubes real quick and label them. Right now there's nothing in them. It is not appropriate to label your tubes after you have made up your standards, because if you get distracted, if the phone rings, if you have to go and talk to a colleague at any point, you can get confused as to what is what in terms of your standards. So I'm just going to label these real quick. 0.625. Okay, and each one I'm labeling with the actual concentration so that I know later what that is. Okay, so let's pop these open again. Right. So now I need to get 100 microliters of this one. So again, my yellow tip is on securely. I'm going to lift my tube up. Again, pipetting 100 microliters, it's a small volume. I want to see the tube that I'm pipetting from. Typically, I'm holding this a little bit lower than you normally would. You should probably hold this closer up to your eye level so that it's comfortable to look at. You're going to want to hold the tube, sort of like, you can hold it like you're making an OK sign so that you can see through the tube here. You can see the pipette tip in there. So we're going to take out 100 microliters. And I can see that my pipette tip is below the surface here. If I left this in here to pipette from, it gets much more difficult to see that I'm actually getting liquid the entire time. You'll also want to watch the speed at which the liquid is being aspirated into the tip. If you go too quickly, and this is usually a problem with a P1000, but if you go too quickly, you may get air bubbles that develop here, and that's obviously going to cause big inaccuracies in your volumetric measurements. Another thing to watch out for, guys, is don't dip your pipette tip all the way into this tube. You can see this tube maybe has, it's quite full. Okay, so I do not need to bury this pipette tip in here in case, that's actually a bad thing to do because I will get residual liquid stuck to the outside of the pipette tip, and that's going to cause inaccuracies in my volume. Okay, so I have my 100 microliters here. I'm just going to get rid of any liquid on the outside. I'm going to close this. This is my stock. I kind of care about it, so I'm going to just make sure that that's closed. And then I'm going to pick up my first standard. You'll notice something that I'm doing here actually. When I'm done with one tube, I move it forward a row to indicate to myself that I'm done with it. So now I'm going to pick up my first standard and pipette into it. Again, I'm going to look exactly at what I'm doing here. I'm going to pipette it into the liquid. I can see my fingers are all out of the way of the tube. I'm making that OK sign again. And I'm going to pipette it up and down about 10 times. Pick a number. that you will always use for like pipetting up and down, rather than just saying I'll do it for a while. So I like to do this 10 times to make sure that it's accurately mixed. I sort of lost count there, but that's one of the reasons why it's important to pick a number, because you don't just want to do this 3 times. You may not get adequate mixing. So I feel like that's probably mixed properly there. You want to avoid bubbles where possible when doing this. Bubbles are bad in biochemistry by and large, so you want to try and avoid them or minimize them as much as you can. Once that's mixed, I want to take 100 microliters from this standard now, and I'm going to dilute it by half in my next standard. So again, I'm going to close the lid of that. I'm going to move it forward 1 spot in my rack so that I know I'm done with that tube, and pick up my next one. And I'm going to go 1, 2, 3, 4, 6, 7, 8, 9, 10. Now before I aspirate here, I don't just want to like suck this up, I want to make sure the pipette tip is empty. Now there's a little bit of liquid stuck on my pipette tip here, and I'm not really confident that that's not going to mess up my next measurement. You can use the same pipette tip for everything, but because I feel like there's a little extra liquid stuck in here, I'm actually going to go ahead and change it. And that's fine. Always use a new pipette tip whenever you think you need to. So this is all mixed up, and I'm going to aspirate 100 microliters here. I'm going to close this tube, move it forward, and put this into my next one. And again, pipette it up and down. 10, I got a little bubble in there, but it's just a small one. Make sure my pipette tip is empty before I take my 100 to go into the last standard tube. So this is 1, 2, 7, 8, 9, 10. Okay, and again, all the while I'm looking at that tube as I'm pipetting into it to make sure that I'm not introducing too many air bubbles, and that I'm actually pipetting liquid up and down each time. So now I'm done with this pipette tip. Okay, so now I have my standards made up and I have my unknown here. So the next thing that we're going to want to do is create our Bradford assay. The Bradford assay is created in cuvettes that will go into the spectrophotometer. Your standards will always be made up in microfuge tubes. So don't do any dilutions in your cuvettes, and on the flip side, don't ever be putting a Bradford reagent into your microfuge tubes here. Okay, let's cap that so that it doesn't get knocked over. Right, so we're going to start off by labeling our cuvettes. The cuvettes that we need, we've got a 5-point standard curve, so that's 5 cuvettes that we will need. We're also going to need a blank, right? For every assay we're going to need a blank. And then I have my unknown protein sample here that I am trying to quantify, so that's going to be 7 cuvettes that I need, which I have here. So I'm going to go ahead and label these cuvettes real quick. So, and you don't need to label them. I mean, labeling it 1 for 1 mg per mil is sufficient. You do want to watch how you're handling cuvettes and where you are labeling them. The cuvettes are obviously going to be analysed by a spectrophotometer. The light needs to pass through the cuvette. If you have written all over the front of the cuvette where the light needs to analyse, the light isn't going to pass through to your sample. It's just going to hit the, the Sharpie line or whatever. So just pay attention to that. So I've just written the numbers on the top here, and you'll notice I'm trying to only handle the cuvettes by the top so I don't get fingerprints on the cuvette, which will interfere with the light absorbance. I'm going to do a big B for blank, and I'm going to do GFC for my gel filtration fraction here. Okay, that's labeled. We'll get this out of the way. Now I'm going to talk about the blank for a second. The blank, I think a lot of us start off when we're doing bench science by just thinking the blank is water. The blank is rarely water, very, very rarely. The blank, in terms of a blank, sometimes it'll be helpful to think of the blank as a vehicle. The blank should have the same as every other test, except the one thing that you're testing. And so what we're doing today is a Bradford assay. Every single assay will get some of this Bradford reagent, and we'll get a little bit of the standard. The Bradford reagent will react with the protein and will turn a particular shade of blue depending on how much protein is present. The deeper the blue, the more protein that's present. Now, we're not measuring Bradford today, so Bradford must go in the blank. Okay? Further, these standards and our GFC, we're measuring protein, so we need to think, what is the protein currently in? And this protein is in this buffer that we used. So this also must go in our blank. The makeup of our assays is going to be 975 microliters of Bradford reagent and 25 microliters of whatever it is that we're measuring. And so our blank therefore will have 975 of Bradford, and then it will have 25 microliters of our buffer here. So let's start off by pipetting 975 microliters, let's get this lid out of the way, into each of these cuvettes. Sometimes people will want to pipette in the small volume first and then put the big volume in on top of it, and there is some logic in doing that. The big volume will mix through the small volume thoroughly. The reason that we're not going to do that is because 25 microliters is a tiny amount to just be pipetting into an empty container. And so in the interest of accuracy, which is of course a priority for me when I'm doing protein determination, I'm going to put the big volume in first. So I'm going to wind my P1000 here, I'm going to wind this to 975 microliters. So again, first I'm going to think, what does this look like when it is wound to its maximum? When this is wound to 1000 microliters, it's going to say 100 on the display here. That's what 1000 microliters looks like. Then it's really easy for me to figure out what 975 would look like. So it's going to say 0, 9, and then the third digit is actually going to be between 7 and 8, and that's going to be 975 microliters because it's in between 970 and 980. I've got my tips here ready to go. My solution is here, and you'll notice how I've arranged this. The tips are actually on my, and you can just barely see them in the edge of the screen there, that's fine. The tips are on my far right because that's the first thing that I do. The next thing I do is I go to my Bradford reagent, so that's the next thing over. And then I'm going to be going to my cuvettes. So there's a reason that I have these arranged like so. These tubes here, I'm actually going to get rid of these and put them over here because I don't need those right now. This is my priority. So I'm going to make sure that tip is on accurately. Now 975 microliters is a large volume. When I press down, there's actually a lot of press on this. You'll want to make sure when you're aspirating that you control how your thumb is going up. Don't just release it like that because you'll find, and I'm not going to demonstrate it here because what happens is the Bradford reagent will actually shoot up through the tip and may enter the barrel of the pipette, which is of course something that can cause a lot of damage to these pipettes, so we want to avoid that. So I'm going to insert the pipette into, you don't need to hold this like this. When we're pipetting with a P1000 from a tube that has so much in it, and it's a big volume, I don't need to lift this up. I prefer to leave it down here because it's stable. So I'm going to put this in. Again, my pipette tip is below the surface, but I haven't buried it too deeply, you can see. And you don't need to just have it like right at the top. I mean, a centimeter or even, a centimeter and a half below the surface is fine. And then I'm in a controlled manner going to aspirate my volume like that. And I have watched this the whole way to make sure no bubbles arose. And I will not remove this until I am sure that there is nothing but liquid and no bubbles inside my, my Bradford container. Now I'm going to pipette these in here. I am going to touch the pipette tip to the corner of the cuvette. And there's a reason that I do that. I don't just want to drop it down from the center. I'm touching it to the corner. One of the reasons is to steady my hand. You can see here it's kind of hard to keep your hand steady. But the other reason is that by touching it to a surface, that surface will actually draw the last of the liquid out of your pipette tip. And again, that's going to help with accuracy. And then at the very end, I'm just going to push out by like pressing down a little bit more that last drop. Again, I can use the same pipette to pipette the Bradford into all of the containers. Number one, because I'm pipetting the same thing, but again, more importantly, these are clean cuvettes. Nothing else was in them, so I am not cross-contaminating my cuvettes with anything. Okay. 3, 4. A little bit of liquid on the outside of this pipette tip is going to have a less severe effect than if we had that same amount of liquid on the outside of a pipette tip with 100 microliters. If you think that you might have 5 microliters on the outside, that would be 5% of our previous pipetting. But it's only a tiny, tiny percent of, it's only like 0.5% of what we've got here. And so you need to sort of consider the different risks of what you're doing at different points and like what kind of a risk is this giving towards the the error of your assay. So you'll notice I pipetted in the Bradford reagent into everything. A common error that I see students make is they prepare their standard curves and they don't really think about their unknown until much later. You want to make sure that you're preparing everything at the same time. Everything needs to be incubated for the same amount of time, and of course this one lonely cuvette at the side here, this is the entire purpose of why we're here today. Okay, this is just our standard curve. It doesn't on its own serve any purpose. The only reason it's here is because we need it for this one cuvette over here. Okay, and so we're going to go ahead now and start adding our standards. We're done with our Bradford, and that will stain everything in sight, so we make sure the lid is on that. Get this out of the way. I'm done with my P1000 tips. Don't need those. Don't need my P1000 anymore. Now, as I mentioned earlier, I'm going to be pipetting 25 microliters of standard or protein or unknown into each of these cuvettes. So again, I'm going to be setting my workspace up because I'm right-handed, moving right to left. My pipette tips will be here. What I'm pipetting from will be here. What I'm pipetting to is going to be over here. Because I need 25 microliters, I'm going to think, what pipette do I need? I'm going to need my P200. I use my P200 because the other pipette that I have is a P20, and I can't use that for measuring 25 microliters. If I had a P100, I could use that, but I don't. So we're going to wind this to 25 microliters. And so again, when this is wound to its maximum, it's going to say 200. So 25 microliters would be 025. So I'll just wind this real quick. And there we go. And now I'm going to pipette in my standards. It really doesn't matter the order that you do this. These are different standards, they're at different concentrations, so I will need to change pipette tips each time. I'm actually going to move my pipette tip waist over to this side, just so that, again, I can enhance the flow of work across here. It's going to be going from tip to sample that I'm pipetting from, to, and then waist. And then I'm done, and I can come back and start over here again. So, I'm going to put my tip on there. I'm going to start with the highest concentration, so I'm going to get my stock, which was 1 mg per mil. To pipette this, I'm definitely going to lift up the tube. 25 microliters is a tiny volume, so I want to make sure again that I can see through. My fingers are getting out of the way of my tube. I'm going to just put this slightly below the surface and aspirate my 25 microliters. I want to make sure that I'm doing this very carefully. Again, if you've got 5 microliters stuck to the outside of this, all of a sudden that's a 20% error. So you want to be very careful when you're doing this. And again, when I put this back in here, I'm going to move it a row so that I know I'm done with it. So I'm done with my 1 mg per mil, and I'm going to pipette this. And this is really important, guys. You're going to lift up your cuvette, just hold it by the top rather than smudging low down, and make sure that you pipette that 25 microliters directly into the Bradford reagent. And then that last thing I did is I pushed out that last bit, and it created a little bubble here. I know I said I don't like bubbles, but I want to be accurate more than I don't like bubbles. So I'm going to put up with that little bubble there just because I know I got every single bit of that 25 microliters into this. What you don't want to do is just put the 25 microliters in and sort of like hang it from a height. You will not get all of that 25 microliters into the liquid. Anytime you are adding a small volume to a larger volume, make sure that you pipette it directly into that liquid. Now you may notice here, I don't know how easy it is to see, the top of this solution is looking a little bit blue, and the bottom still looks that kind of browny color that the Bradford has when it doesn't have any protein in it. So I'm going to need to mix these at some point. I'm going to mix them in a little bit here. Don't try and mix them by pipetting up and down with your with your pipette here. It's only wound to 25 microliters, and that's not sufficient for good mixing. So we're going to come back and actually mix those properly later. And again, you'll notice I'm going to move my cuvette a row down so that I know that I'm done with that one. So we need to change our pipette tips. We're going to get, and I'll move pretty quickly through these last ones, or these remaining ones, 25 microliters. In here. And you'll notice because I have my workstation set up somewhat conveniently, you can actually move pretty quickly through these. Notice that my pipette hand is actually barely moving. I like to keep that pretty still because this is where the accuracy is going to come from. I don't really like to move that too much. I'm going to move this tube to it. I'm going to move the cuvette to it. Whoa, that doesn't go in there. Put this in here. Final one. And each time I'm doing this, I am pressing down that last little bit. You'll notice when you're using a P200 to pipette 25 microliters, there isn't actually a lot of give in that plunger there because 25 microliters is a small volume on this. So you just press it down a little bit. Make sure you don't accidentally go all the way down to the bottom. I do press when I'm dispensing, I do press out that last little bit by pressing out that last little bit, but just make sure that when you're aspirating your sample from here, you don't go all the way to the bottom. And now I've pipetted 25 microliters in here. I'm going to go ahead and immediately put my unknown in here as well because it needs to sit the same amount of time as my standards. 5 microliters. That's my unknown. And everything is labeled, so I know what everything is. We're going to move this to the same row as the other ones because it's now done. And I notice here that I haven't done anything with my blank. Now, as I mentioned, I'm going to have to put something into my blank. So because all of my samples are made up in a Bicene buffer, that's going to be what I use to add to my blank here. So I'm going to add 25 microliters of Bicene. And I'm going to see, you might not be able to see too clearly, but I'm making an effort not to dunk my pipette tip into the Bicene buffer, again, just so that I don't get any extra liquid on the outside. Okay, and now the blank is done. So I pipetted everything in there. My pipette out of the way, we can close our tubes, we want to close our pipette tips, we sort of want to keep these clean. The next thing we need to do is mix these. You can see just by looking at your samples that these are not mixed. Okay, so I encourage you, take a second, take a quick look. Sometimes you'll want to actually pick these up and sort of look through, like with a light behind, you'll be able to see really easily that they're not mixed. Now, there's 2 ways that I'm going to recommend that you can mix these. One is using a P1000. So again, that's going to be a large volume. Or what's okay to use is just like a transfer pipette, which is just going to aspirate and release. And again, when you're doing this, you just want to make sure that you minimize any bubbles that are formed. If you have a few, that's okay, but you just don't want to be frothing it up. The blank I'm going to mix first. I can actually use the same transfer pipette to mix all of these if and only if I go from the lowest concentration to the highest concentration. So I'm going to start with the blank, which is obviously the lowest concentration. There's no protein in there, it's just Bradford and buffer. So I'm going to mix that, and it actually, I mean, there's no blue in here, obviously, I just put buffer in here. Now you want to make sure that you don't like include bubbles. So you're just going to gently pipette it up and down, and it doesn't take too much to see that it's done. You will want to make sure, because I plan on using the same transfer pipette here, that I have actually released all of the liquid out of this transfer pipette. And I'm going to do the same thing, I'm just going to move it a row to show that I'm done. So I'm going to move this back up. I'm going to do the next lowest one, pipette it up and down. And make sure these transfer pipettes take a little bit of getting used to. You want to make sure you squeeze the bulb before it goes into the liquid. Obviously you're just going to be blowing bubbles if you don't do that. And it's really easy to see if these have been mixed properly. If they haven't been mixed, you know, when you look at them through the light, there's going to be like little blue tinge to the top. Okay. And our last one here is going to be this one. So now my standards are all mixed. I can see that they are mixed well. I am not going to use this transfer pipette on my unknown. I have no idea what concentration this is, so I can't put this in here. Always use a new mixing, like whether you're using a pipette tip or a transfer pipette, always use a new one for your unknown. So we're going to mix this up and down gently. That is done. And you'll notice I'm throwing everything away when I'm done. You don't want to be leaving used pipette tips or used transfer pipettes or anything on the bench. Equally important, something that I didn't mention, is when you lay down a pipette when you're not using it, make sure that there is never a pipette tip on it when you lay it down. Okay, so let's have a look at our standard curve here. So I'm looking at this now, and one of the things that I want to watch for is, first off, there should be a smooth gradient of color in my standards. So going from low, the blank is pretty much just Bradford on its own with a little bit of buffer. And this, you know, it looks sort of like a little bit of like a goldeny brown when it has no protein in it. And so what I should see with my standards is going from very pale up to a nice blue color in a nice smooth gradient. If you notice that any cuvettes look the same as each other, or if there looks like there's a bit of a jump from one color to the next and that it's not smooth, that indicates one of two things. You either made an error when you were creating your standards over here, or you were not being accurate at some point when you were adding 25 microliters in here. The next thing that you want to watch for is that you don't have a super blue for your top one. This top one is a 1 mg per mil solution, and the way that we've made this up, with 25 microliters of standard going into 975 of Bradford, it shouldn't be too much of an intense blue. So you can see here, I mean, I'm not really sure how to describe this shade, but when you get like a royal blue, a really deep royal blue, sort of like the color of my top, then that's going to be too blue. Okay, I've just closed my lab coat here, which probably should have been closed all along, to be honest. But this is going to be extra beneficial, not just to protect me, but also you guys can see the colors a little bit more effectively maybe here. A couple of things that you're going to watch out for once you've prepared your Bradford standards. The first thing, to be honest, is probably you want to have a look at the volumes in each cuvette. I should have 1 mil in each cuvette. Okay, and so if any of them looks a little bit lower than another one, that's going to be an indication that either I forgot to put something in or I wasn't being careful when I was pipetting or something like that. The next thing, we're going to look for a smooth gradient as the protein concentration increases from nothing in our blank through our standards here. It should be smoothly increasing, and ideally your lowest concentration should be distinguishable from your blank. And it's a little bit hard to see here, but if you just make sure when you can just pick those up, compare them to each other, and yes, I can see here that my lowest concentration does have a darker color or a little bit of a blue tinge to it compared to the blank, and then I can see that there's a smooth increase in color here. I want to have a look at my strongest, my highest concentration here, my 1 mg per mil. So this is a nice shade of blue. You don't want this to be too much of a royal blue. Anything too close to the writing, for example, on the cuvettes here is going to be too blue. And the reason for that, of course, is, as we've discussed in class, the linear relationship between absorbance and concentration only holds for certain concentrations. Once you get any more concentrated than this, it starts to break down and it becomes more of a plateau. Okay, and then finally we want to have a look at our unknown over here. And of course this is the entire purpose of why we're doing the rest of it. Okay, so we've got these 6 tubes here. They just serve the purpose so that we can compare this one. And what we want to do is make sure that the concentration of our unknown falls somewhere within our standard curve. Now, rather than wait the 10-minute incubation time that we need to before we can assay these, what I can do is I can just look, I can just hold this up and sort of compare the concentration, just compare the shade of color. Okay, you should be able to tell if it's bluer than your bluest standard. If it is, that is too blue. It is not appropriate to extrapolate values from your standard curve. Your unknown must always be read from within the range of measured standards. And so I can see here, this is a little bit on the high side, but it's definitely not as blue as my bluest standard. So I'm thinking it's actually somewhere in between these two, and that's going to be fine. And you don't need to spend too much time just looking at it. You just want to hold them up and compare. And then I'm pretty happy with that. Now, if it had been bluer than this, we would have had to make a dilution of our original GFC fraction over here. And typically, if it's going to be too concentrated, make sure you do a meaningful dilution so that you don't have to repeat your work. Just make sure that you go ahead and do a 1 in 10 dilution immediately rather than doing like a 1 in 2 dilution, because you could end up having to just repeat that dilution needlessly. And so now our standards are prepared, our unknown is prepared, we have our blank, we're sure that everything is made up properly, and so we just need to incubate this at room temperature. So I'm going to just leave it sitting on the bench for 10 minutes, and then I will assay it on a spectrophotometer. Okay, so now guys, I am going to demonstrate how we're going to assay our samples using a spectrophotometer. The spec that we will use to assay our Bradford assay is going to be a SpectroVis Plus, or we've been calling them mini specs in the lab. These are just tiny little visible spectrophotometers. They can shine light of any wavelength in the visible spectrum. And our samples are blue, therefore they will absorb light in the visible spectrum. The lambda max, meaning the wavelength at which Bradford will maximally absorb, is 595 nanometers. And review the visible spectrum and how spectrophotometers work if you need to, but you should be able to make a reasonable estimation of the lambda max of any colored molecule. So the first thing that we're going to want to do is open up our Logger Pro software. The Logger Pro software, I'm not going to show opening it here, but Logger Pro software is going to be on the desktop of any of our lab computers. So just go ahead and open that up, and you'll see the screen. It will look a little bit something like this. Now the fact that this screen is white tells me that my spectrophotometer is not connected. And so I'm going to go ahead and plug in the spectrophotometer to one of the USB ports on the computer. And I should see this, once it connects, I should see a change on the screen here in a sec. Let's just give it a sec to recognize the spec, and this is what we're going to see. This looks a little bit odd here in the middle. You can guess the color that is going there in that strange stripe. When the spectrophotometer is connected to the computer and you open up Logger Pro, you will see a rainbow across the middle of the screen. That's how you know you're connected. If it shows white like we had a second ago, that's just indicating there's no connection between the spectrophotometer. And so you just want to check the connections if that's the case. Now when we open up when we are ready to start assaying our samples, the first thing we need to do is calibrate our spectrophotometer. And so we're going to do that by, I'm just going to move some things out of the way. We're going to do that by going to, I'm just going to move this across. I have gloves on here, so the mouse is moving slowly. We're going to click on Experiment first. Here we go. And we're going to drag the cursor down to Calibrate. We want to calibrate our spectrophotometer. This is going to be really important, and you will need to calibrate the spectrophotometer each time before you use it. When you click on that calibrate button, you're going to see this dialog box. It's initially going to warm up the spectrophotometer. This warm up is important. There's a lamp in the spectrophotometer which is going to be shining light at your sample. If the lamp is warming up while you are assaying, then the intensity of light that it shines at your sample will be different as you move through your samples. And that will just lead to inaccuracies in your data afterwards. So you just want to make sure that you go through this warm-up here. Now this spectrophotometer has previously been warmed up, so I'm going to go ahead and skip the warm-up. If you've been using the spectrophotometer a lot and you've just recently connected into the software, it's okay to skip the warm-up. But if you haven't used it yet in this particular lab session, you must complete this warm-up. The next thing that you're going to see when the light warm-up is done, you'll see this finish calibration here. It will not finish the calibration until you press this button, and it's giving you a prompt over at the side. It says place a blank cuvette in the device. Now what it's looking for here is your blank. So what you're going to do is you're going to take your blank Bradford sample and you're going to place it into the spectrophotometer. Now when you do this, make sure that you line up the arrows on your cuvette with the arrows on the spectrophotometer. You need to make sure that the light is shining through the spec in the correct direction. All sides of the cuvette are not optically equivalent, so make sure you line up your arrows correctly, or again the light is not going to be able to travel optimally. So my blank is in the cuvette now, so I'm going to go ahead and click Finish Calibration, and this just takes a few seconds here, and it's going to look at my sample, and essentially what it's doing here is it's looking to see what does nothing look like. For spectrophotometry, all your values are always just relative to each other, so you need to start off by showing it what 0 looks like. And I know that I have 0 protein in this sample, so that's what I've just done. I've just told the spectrophotometer what 0 looks like. So now that that's done, I'm going to go ahead and click OK. And now my spectrophotometer is calibrated. The next thing that I need to do is configure my spectrophotometer. So there's two things essentially we need to do before we read our samples. The first is calibrate, which we just did, and the next one is configure. So to configure the spectrophotometer, before I start that, I need to put a sample that has protein into the spectrophotometer. And usually I use one like maybe my 0.5 mg per mil standard. So I know that it definitely has some blue color to it. And again, when I put this into the spectrophotometer, I'm making sure my arrows are lined up with the cuvette and the spec so that the correct part of the cuvette is being exposed to the light. And then I'm going to move up to the top toolbar here, and I'm going to click on this button here that looks like a rainbow. And you'll see there, that's the configure spectrophotometer button. Now when I click that, it's going to open up this box here. So we'll go down and we'll just go through these one by one. The collection mode. The collection mode that we want to use is absorbance versus concentration, because that's what we're doing. We're creating a standard curve. So I'm just going to go ahead and just click that. You'll notice here that these boxes have opened up for editing. This one looks okay. Our short name is fine. Let's have a look over here at our units. Now this says moles per litre. So this is not my concentration. If I think back to what my standard concentration was, it was 1 mg per mil. So my units of concentration are in mgs per mil. So I'm going to go ahead and click on this, and then I'm going to delete this, I think. Here we go. And I'm going to put in mgs per mil. There we go. That's the units of concentration that I'm using. And then you can go ahead and let this auto-select it at a single band here. The next thing I'm going to do is have a quick look at my, my absorbance spectrum here. Now what this graph is showing us is basically down here on the x-axis we have all of the wavelengths of visible light that were shone at our sample. So we can see that there is no absorbance for the lower wavelengths of light, and that the maximal absorbance happens up here at the top of this peak. This is why it's really important, guys, that you've got your an actual blue sample in the spectrophotometer when you're configuring. If you put your blank in here, all you're going to see down along here is a massive zigzag mess, because it doesn't absorb maximally. One of the main purposes of configuring the spec is to determine the lambda max. You'll notice there you can auto-select the lambda max if you want. This would be an inappropriate selection where I have here, because I'm on the side of the curve. You always want to analyze all your samples where they maximally absorb light. And so we're going to go over here and we're going to just manually select 595 nanometers. Now it probably had this like auto selected up here if I go like that. That's around about where it was initially. Where it auto selects, you may leave it at the auto selection, but I like to look at what I know the lambda max for the molecule is. So it's 595, and this here 594.8 is the closest, and I'm just going to have a look at that, and that's still like right there on the top of the curve. Whoops, I just changed that. This is a sensitive little thing. Probably should have a mouse instead of the trackpad. Let's see, 594.8. Okay, let's steer clear of the graph so that we leave that selected. You see how easy it was there to just change that inadvertently? So just, you know, exercise some care when you're going through that so that you've got the correct wavelength selected. Once that's all done and you're happy with that, you're going to go ahead and click OK here. Okay, and then you're going to see a white screen. So to recap what we've done so far, we turned on the Logger Pro software and had the spectrophotometer connected. There's two things that you need to do. The first one is calibrate. You do that with your blank. The next one is configure, and you do that with one of your samples that has color. Okay, and then you're going to see this screen here. So now we can start acquiring our samples. At any point, if you want to know the absorbance, have a look at the lower left corner with the red writing here. This is going to show you the actual absorbance of your sample, and so we can see here for this one it's 0.628. Now I like to acquire my samples in order of concentration, so I'm going to go ahead and start with my lowest concentration sample, which is my 0.0625. Something to note here, guys, you don't need to acquire your blank. You've already set your blank to 0, so there's 0 point, if you will, of putting your blank in here and acquiring it. So I'm going to put in my 0.0625, and the absorbance, again, as I mentioned, is going to be displayed down here. And when you put your first standard in, you're going to go ahead and press collect. And what you'll see is on the screen, you'll notice there's a red dot has appeared down here. Okay, that may move when you first press collect. If it continues to move for more than about like a second or a half a second, that indicates that you probably haven't mixed your samples. So just take out your sample from the spectrophotometer and have a look, because again, as I mentioned earlier, if your samples aren't mixed, you can usually see it pretty easily. Now once that's sitting there pretty happily and it's not changing too much, you're going to press keep. Okay, so when I press keep here, a little dialog box will pop up and it's asking me what's the concentration. So I'm going to go ahead and type in the concentration. It's 0.0625. And you can see the units are auto entered here. It's mgs per mil. I'm going to go ahead and press OK on that. Here we go. And there's my first point in my standard curve. And then you're just going to continue with the rest of your samples. So our next one we'll put in here, and you see the dots appear one by one. And so one of the important things to keep in mind here, guys, is don't press stop until you are finished with all of your standards. We're going to press keep instead for each one. And so this one is 0.125. There we go. We're going to do our next one. And you'll notice that it's automatically plotting the points for me. This is 0.25 mgs per mil. That one is not looking quite perfect as I would like. We've got the next one. Press keep on that, and this is 0.5 mgs per mil. And then our last standard, we'll keep that, and this is 1 mg per mil. So here's my standard curve. So just on a preliminary look at this, it's not bad. I'm thinking maybe this is a little bit of a blip. You know, in fairness, this is a pretty good standard curve. I'm just, you know, really just picking at details here, but it's not perfectly straight. This point should probably be a little lower. That might indicate a little bit of inaccuracy when I was adding the 25 microliters to the Bradford earlier. Probably constant talking when you are preparing your assay is going to lead to a little bit of inaccuracy. Now, I've collected all my standards, so at this point I can press stop. Okay, so I have all my standards. It is really important that you do not acquire your unknown as part of your standard curve. Your standard curve is for standards that you know everything about. So I know the concentration and I'm measuring the absorbance. I don't know that about my unknown, my GFC fraction, so I'm not going to acquire it as part of my standard. Now once I press stop here, I can see that my standard curve data is over here. The concentration is here, the absorbance is here. Typically at this point, what we're going to do is export this data to Excel. You guys don't have Logger Pro on your personal laptops at home. We're much more used to handling our data in Excel, and so we can just take it straight out at this point. But before we do that, we are going to have a look at the concentration, or rather the absorbance of our unknown sample, because of course that's the entire reason for everything that we're doing here. And so we're going to pop that into the spectrophotometer, and all I'm going to do at this point is just look at the absorbance that is displayed down here in the lower left corner, and I'm going to record that. Okay, it's 0.0, around about 0.08, and it's fluctuating there a little bit. That's perfectly normal for it to fluctuate in the third digit. Don't worry too much about that, as long as the first two are somewhat stable. And so that's going to be the concentration of my unknown. And again, I can see that or the absorbance rather, the absorbance of my unknown is well below the absorbance of my highest standard, which of course is really, really important. Okay, now to export this data to Excel, there's two ways that you can do it. The first thing that you can do is you can go up to File, and you can go down to Export, and you want to export as, and you can export as a CSV file. Okay, so just make sure that you save it and make sure that you label the file accurately so that you'll be able to find it in your folder on the desktop. The other thing that you can do, if you're already working from an Excel workbook, you can just select this data like this and just copy it and paste it directly into an Excel workbook. Okay, so if we just open up Excel here, I can just go ahead and paste this directly in here and all my data is there. Now you'll note it's just put in the numbers, so you must of course make sure that you've got your columns labeled. Now, let me just go ahead and label that. I don't like unlabeled columns here. If I get distracted, I'll come back and not know what these are. So concentration in mgs per mil. Did I put that in correctly? No, I didn't. This is hard to do when you're...
Alright, start over, you said shit.
Okay.
Go back to where you said you were going to label your columns.
Okay. Okay, so we're going to go ahead and label these columns just to make sure that we don't get distracted and forget what they all mean. So concentration is what goes in here. You can abbreviate that with just conc if you like. Concentration in, whoops. Concentration in mgs per mil. Make sure, guys, when you are labeling columns like this that you include the units. That's really, really important. So this is going to be absorbance. So we can abbreviate absorbance to ABS if you like. Include not just absorbance though, but absorbance at 595 nanometers, okay? I do like to know, because you can measure the absorbance at a whole bunch of different wavelengths, so I do want to know what that is. Let's just make this a little bigger here. Now I can actually draw my standard curve that I had in here. So I'm just going to insert a graph, and again it's always going to be when you've got X, Y values, you're going to be inserting a scatter graph. I usually like to insert one that already has a line so that I can see it. And there's my standard curve. So you're going to want to make sure, I'm not going to go through it here, but you've heard me say it enough in class, you're going to want to make sure that you label your axes appropriately. Let's just have a quick look at the R squared value. We're going to display the equation of the line which we need and also the R squared value here. And that's pretty close to straight, and we've got an R squared value of 0.999. Certainly you can aim for that, and I can see here this little blip that I would normally expect to be more in line with the others. And that's what you're going to be aiming for, guys. In terms of your exam where you're going to be doing this, I'm going to expect you guys to have an R squared value of at least 0.97. Okay, so that's definitely going to be your target, and again that will be controlled solely by how much attention are you paying to your pipetting. So we have the equation of our line. Back at Logger Pro, we had the absorbance of our unknown here, so we will record that 0.68, and we're just going to insert this over here. We're just going to just record this here, just unknown, or maybe I'll call it GFC actually, because that's what the sample is. So GFC, and then the absorbance of that was 684. Okay, now all of my data is in here. Okay, and so I can use the equation of my line to determine the concentration of my gel filtration fraction here. So I have a Y value, that's what I determined, my absorbance is a Y value. Therefore, I can determine what the concentration is, the X value, by just filling in for Y and solving for X here. Okay, and again, I'm not going to go through it here, but make sure that you label axes with what they are and their units. Okay, so we are going to leave it at that. Once you're done with everything and you've acquired all your samples, You must of course remember to clean up your workspace. So that's going to involve getting rid of your samples. One thing about Bradford reagent that we've mentioned, it cannot go down the drain, so just make sure that you put your Bradford reagent, you empty your cuvettes into the designated Bradford waste. Most other things can probably go down the drain, but make sure that you seek clarification from either myself or your TA if you have any questions about where things can go. And that's all we've got. So you guys should be able to pipette accurately and also determine the concentration of an unknown protein by doing a Bradford assay.